Hey guys, this is going to be a fairly short video about meiosis and sex cell production. I want to clear up some things about how meiosis works and how it's different than mitosis. If you remember way back, mitosis, when you get into mitosis, the purpose, the end goal of mitosis is to have two identical cells when you started off with one cell. You want those cells to be exactly the same, perfectly reproduced. You have to imagine it like skin cells. Your skin is constantly sloughing off. You take a shower, it sloughs off. You rub against your sweater or your coat or something, and skin rubs off. And you're constantly replacing it, but you don't want to replace it with a different cell, right? You want exactly the same kind of cell. That's the purpose of mitosis. Meiosis is not, that is not the end goal of meiosis. The end goal here is to develop gametes. And in men, those are, I got to get my little pointer thing here. Right? In the male sex cell, the gamete is called a sperm. And in the female gamete, it's called an oocyte, or an egg. Now, it's actually really called a secondary oocyte, but we'll get more particular about that later on. Also, the purpose of sexual reproduction is to have the sperm has half of the genetic complement that you need to create that being, to create the human. And the egg has the other half, right? So most of the cells of your body, what we call the somatic cells, S-O-M-A-T-I-C, the somatic cells of your body, then that's most of the cells of the several trillion cells that you have. Most of those cells have 23 pair of chromosomes. That's 46 total chromosomes, but 23 pair. And remember, they're a pair because you get one chromosome number 1, you get one of those from mom, and you get the other one from dad, and so you have two chromosome number 1s, but one is from mom and one is from dad. And then you have two chromosome number 2s and two chromosome number 3s, and so on. And so that gives you 23 pair of chromosome. Of course, then we get into that, well, that's 46 total chromosomes. Well, yes, it is. Right here. You get 46 total chromosomes. Yep. But the point of meiosis and the point of creating a gamete is to create one chromosome of the 23 in a sex cell. So the sperm has 23, the egg has 23. Now, when you put them together, you get 23 pair. All right. So that's the end goal of meiosis. The other end goal of meiosis is I told you that mitosis, the end goal is to get identical cells, no variation. The end goal of meiosis is to get variation. You want variation. That's how evolution continues, by having variation. Meiosis is that method. So now I've added a couple slides to the slideshow that you have. We got to go through some terminology here, and it gets confusing. So every cell in your body has, let's say this is chromosome number 1, okay? There's chromosome number 1. You have one of those from mom. You have one from dad. So that would be a pair, right? In the somatic cells, you have a pair. Here's chromosome number 1 from mom, and here's chromosome number 1 from dad. We refer to those as homologous pairs. Homologous pairs doesn't mean they're exactly the same in every single detail. Of course they're not, because one came from your mom, one came from your dad. But they're very, very similar. They have the same genes on them, although those genes can be slightly different from one another. But we refer to that as a homologous pair. Okay, now before you go through either mitosis or meiosis, you have to double up the chromosomes. So you have to double this chromosome and double that chromosome, right? So you get 2 of those and you get 2 of these. And that's in preparation for replication. And that's also in preparation for meiosis as well. You have to double up everything. What you get, so if this chromosome doubles up, what you get is that. So this is a duplicate. It says over here that's a duplicated chromosome. So this chromosome has been duplicated. Now, here's where we get into weird name changes that will throw you off sometimes. We referred to this one thing down here as a chromosome, and it is. But when we double it, its copy, here, this we call that a chromosome, and we would call this a chromosome, and we would say, oh, we have 2 chromosomes. And well, what we say is, if they're connected at the centromere, these are identical. We call them chromatids now. So we've changed the name. So they're called sister chromatids. Sister chromatids are identical copies. So if you started with this chromosome, or, same thing here, this one, if I double that one, I get that. Oh, that's a terrible arrow. I get that. I'm going to do the same thing. It doesn't show you that, but here I'm going to double this red one too. I'm going to get the same thing. I'm going to get 2 red ones connected together, and we're going to call them sister chromatids. And the sister part has nothing to do with whether they come from male or female cells. Has nothing to do with it. It's just because they are exactly the same. So you have sister chromatids after you have doubled up the DNA, after the DNA has gone through the replication cycle in preparation for either mitosis or meiosis. Either way, you have to double up the chromosomes. So you have sister chromatids. Okay. Now we're going to move on. Now, meiosis, this is where meiosis differs from mitosis. In meiosis, you're going to take, these are the double up chromosomes. So we're saying, in this particular case, we're saying this one is the doubled up maternal. So the maternal chromosome, that comes from mom. And they're labeling this one as the doubled up chromosome from dad. So it's replicated. You've already replicated the DNA, and you have sister chromatids, right? So the maternal and the paternal members of each homologous pair are positioned parallel to each other, right? So now they're sitting next to each other. Even though they're identical copies, now, here's the magic of meiosis. The magic of meiosis is this thing that's called crossing over. Crossing over is these 2 pairs that have been replicated, they get real close to each other. And at this point, this is called a tetrad. And tetra means 4. So you've got 4 individual chromosomes, and they do this magic thing right here. They get real close to each other, they get next to each other, and they swap genetic material. That's a crazy, weird thing. They swap genetic material. Now, they are no longer identical pairs. Right? So, what you get out of this is crossing over creates variation. Variation in chromosomes at the end of metaphase 1. So in metaphase 1, the whole point here is to create different to swap genetic material so that you get different chromosomes out of this, and that's where variation comes from in reproduction. That's why your parents can have certain traits, and you have maybe some of those traits, but you may have different traits. That where did they come from? Well, they came from the swapping of these parts of chromosomes. And that whole thing is called crossing over. It happens before you get to metaphase 1. Then when you get to metaphase 1, metaphase 1 will cleave this thing right down the middle. So this chromosome, well, this pair of chromosomes actually heads off that way. This pair of chromosomes heads off that way. And at the end, so this is what you get right here. This is what you get at the end of metaphase 1. You've got the crossed over chromosomes, but they're not ready to become gametes. Then you go through the process. Now, they show you metaphase 1 here, and they show you metaphase 2 here. Okay? Well, you have to go through all the other phases, right? You have to go through, the way to remember it is PMAT, PMAT, right? You have prophase, metaphase, anaphase, telophase. So you have to go through, P would be up in here, and there's M, and that would be P1. Ew, that didn't work out very good. This is M1. Then you have to go through A1 and T1. 1. And then, in meiosis, you get this next phase called metaphase. Metaphase 2. So they go through the whole process again. Prophase, metaphase, anaphase, telophase. But the whole second part of meiosis resembles mitosis. It's much more similar to mitosis. But the first phase does not resemble mitosis because of this whole thing called crossing over. In the end, what you get down here is gametes. So we talked about in females, it's egg cells. In males, it's sperm. So what you get is each one of these has a different combination of genes. The same genes, but different combinations of them because they swapped over way back before they're way up in the crossing over phase. So you get 4 unique gametes that you can reproduce with. Alright, so this whole slideshow goes through the whole process. Now, these, when this slide says replicated chromosomes, these chromosomes are replicated and they have swapped over. And you can see that. I don't know if I can magnify it, but you can magnify it. Oh yeah, maybe I can. Aha! Hey, look at that. I can magnify it. Look at me. Alright, so you see, these chromosomes have swapped genetic material. So they have already done the crossing over thing. So then, now, in metaphase 1, these chromosomes, these replicated chromosomes, are going to line up. And you see this middle line, this equator. You get a pair of chromosomes on one and a pair of chromosomes on the other. So remember, these are all, we'll say this is chromosome number 1. Well, I've got 4 chromosome number 1s. But the homologous pairs line up on one side, the homologous pairs over here line up on the other side, swapped over homologous pairs. They line up like that, and then they go in their separate directions in anaphase. So, at the end of telophase and cytokinesis, what you get is you get the replicated crossed over chromosome in one cell. So all of these ended up in that cell, and all of those ended up in this cell. Now you have something akin to mitosis. Now you've got to take apart those centromeres. You've got to take these chromosomes apart from each other. And we will come down to, oh, that's the next slide. Haha. I have to get out of the, I think I have to get out of the, oh dear. Yeah, I'm stuck now. Wow. How do I get out of this? Ha. Hey, I'm back. I figured out how to get out of the magnification thing. Okay, so now I'll go to the next slide. You've ended up at the end of, this is all meiosis 1. See that up there? Meiosis 1 is up there. Meiosis 1 ends in 2 cells with replicated crossed over chromosomes. Now we go into meiosis 2. So each one of those cells that you formed from meiosis 1, now each one of those cells now has to go through its own division, and now you get something akin to mitosis. See this this equator line now, that line there. Now those centromeres are lined up right on the equator, just like they would be in mitosis. And each of these sister chromatids now separates and goes its separate way. Right down that middle. This one too. So this is exactly the same. So this is just like mitosis. What you end up with is daughter cells that are considered to be haploid daughter cells. And so now these are gametes. So you have 1, 2, 3, 4. 1, 2, 3, 4 gametes. Now you're done with meiosis. Now, in sperm production, these things have to develop tails and they have to become sperm. But now they're genetically ready to become sperm. In females, they have to become eggs, but it turns out that only one of them actually becomes the egg for fertilization. The other 3 don't make it. There's a whole process to that. We'll get to that. So now here's your slideshow, okay? So meiosis includes 2 successive divisions. You've seen that now. The first one's called meiosis 1, and everything is referred to as a 1 in that. The second one is meiosis 2. Prior to meiosis, you gotta replicate the chromosomes. Now, and I've told you how they become what are called chromatids then. What you used to call a chromosome You replicated it. Now you have sister chromatids. They're connected by a centromere. Those cells are diploid cells, right? Because you have two sets of chromosomes in 23 pairs. In meiosis 1, you separate the homologs. If you separate the homologs, then you have what are referred to as haploid cells. A diploid cell has two homologous chromosomes in the same cell together. You don't get that out of meiosis 1. What you get is the homologous pairs separate. So they're called haploid cells. And as you see, the definition, where'd my little pointer go? There it is. The definition of haploid, right, contains one set of chromosomes. These chromosomes are replicated chromosomes, and each one of those contains a chromatid. The terminology is kind of weird. And then you go through the whole process of meiosis 2, which separates the chromatids, and then producing cells that are still haploid, but now each cell in the end has one chromatid. So here's your book. Now your book doesn't show you the fancy crossing over stuff, because they don't give you any different colors. They have everything's the same color here, and I find it somewhat confusing. You miss the crossing over. Crossing over is an essential part of that, and it's already occurred. But this is the same process that's in your book. Oops. So the first phases of meiosis are the same as mitosis: prophase, metaphase, anaphase, telophase. It's just that in meiosis it's all referred to as one. Prophase 1, metaphase 1, anaphase 1, telophase 1. You can read through what happens there. This is your book showing what happens. This is crossing over. Right, so by the time you get to metaphase 1 in meiosis, this has already happened. So these are the crossed over ends of chromosomes or parts of chromosomes, and you can get more than one. They just show one crossing over. You can get double crossovers. There are lots of different ways that these things can cross over, but we try to keep it simple to show you just one crossing over point. What you get is recombination, of course, as it says here, and the results, you get different genetic combinations, and it ensures that each new organism is unique. That's the point of procreation. That is the point of evolution. This is how it occurs. Now, second meiotic division pretty much heads along similar to mitosis at this point, and you can go through the phases there. In the end, here's what you get. You get 4 gametes. They each have different genetic variation. Remember I said, in eggs, only one of the egg cells matures, and the others degenerate. Division ends with each sex cell having one set of genetic instructions, or 23 chromosomes. So each genetic cell has 23 chromosomes. But in order to make another human being, you have to have 23 pair of chromosomes. Now this is your book showing crossing over. Now they have color-coded, color-labeled things. And so you can follow that through, but you know what crossing over is now, right? So, and they're showing you, oh, different blood types. You know, here's a blood type gene. Here's an eye color gene. Here's a hair color gene. And maybe some of those flip-flop. These are maternal chromatids. And the paternal chromatids. So, you know, so they're saying, well, your dad had a certain color hair and your mom's got a different color hair. And you may end up with some variation of that because you get crossed over chromosomes. And then in the end, the gametes, the 1, 2, 3, 4 gametes that you have, are going to be, some of them are going to be quite different than the parents. Some will be fairly similar. But that's how evolution works. Alright, that's it for this. That's all I wanted to go through. I hope this helps to understand meiosis a little bit better.